Cells with the same genotype can look and function differently. My nerve cells look and function differently from my bone cells, and the older me is wrinkled and grey. Yet the DNA sequence is generally the same in different cells of my body and as I age, give or take a few mutations. Different phenotypes arise in cells with the same genotype when different genes express different proteins to carry out different functions in each cell type. A factor that determines whether or not a gene expresses its proteins is epigenetic modification. The epigenetic modification includes the attachment or removal of molecules to DNA itself, not changing the base sequence, or to the histone proteins around which DNA is wrapped, and the positioning of the nucleosome bundles. DNA methylation could within a promoter region recruit gene suppressor proteins. Methylation also drives heterochromatin formation, so the nucleosome tightening prevents transcriptional machinery from interacting with the DNA. Histone modification may occur through acetylation, methylation, phosphorylation, and ubiquitination, which all alter DNA-histone interactions. For example, histone acetylation at positively charged lysine residues weakens the DNA-histone interactions, thus opening the chromatin to form the open euchromatin state, which facilitates gene expression by allowing transcription factors and enzymes to interact with the DNA. Non-coding RNA sequences, too, may be vital in regulating gene expression. The epigenome shows relatively stable inheritance, so tissue homogeneity is ensured. The liver, for example, consists of many cells expressing the same genes and working on the same function. In sexual reproduction, most of the epigenetic layers are removed in the zygote to be re-established in the embryo's development. As the embryo develops, the signal a cell receives from its neighbors helps determine what type of cell it will become, so the removal of epigenetic tags of the parents in the zygote enables the different cells and tissue types needed for life to develop. However, even in sexual reproduction, some epigenetic tags are passed on through the gametes to the zygote and eventually to the new individual. The inheritance of epigenetic markers from parents to offspring is seen in children whose mothers were malnourished early in pregnancy. Individuals conceived during the Dutch hunger winter in 1944 and 45, even as adults, had less DNA methylation of the insulin-like growth factor 2 gene than their unexposed same-sex siblings. They also had a higher incidence of metabolic disorders, and these effects seem to be present in the children of this group, which is the grandchildren of the women who were malnourished in the first 3 months of their pregnancy. Similarly, exposure to the horrors of the concentration camps affected the methylation of the PKBP5 stress gene of the Holocaust survivors and their offspring not conceived during the traumatic period. The change in methylation of the stress gene caused anxiety disorders and depression, even in the children who were not exposed to the trauma. So factors such as diet and stress induce epigenetic modifications which can pass down from parents to children. Thus, we can conclude that our phenotype depends on our cell's DNA sequence and the inherited or acquired epigenome. Nature and nurture inevitably interact. Epigenetic modifications are reversible, and therefore researchers are looking to advance disease treatment by targeting the epigenetic marks on the genome. In summary, both genes and the epigenome influence our phenotype. The stable mitotic inheritance of the epigenome is essential in development. Most of the epigenetic information in mammals is removed in the embryo to enable the development of different cell types. However, some epigenetic signatures are transmitted from parents to the embryo. The epigenome is susceptible to environmental exposures such as nutrition and stress, especially at specific times in development. Although we may experience long-term effects caused by exposure to our ancestors, we can make choices that modify the epigenetic tags and benefit us and the following generations. Researchers are looking to advance disease treatment by targeting the epigenetic marks on the genome. Here are some references to study the topic further. Now let us check your understanding of epigenetics with a few questions. Question 1. State whether the following is true or false. Only cells in the eyes have genetic information for eye color. The answer is false. Cells, tissues, and organs differ because they have different sets of genes that are switched on or activated and switched off or deactivated. Nearly all cells in our body have the same genetic information. The zygote forms cells of the organism by mitotic cell division. Question 2. What is epigenetic inheritance? A. An inheritance pattern in which the DNA sequence isn't altered but gene expression is. B. An inheritance pattern in which the DNA sequence is passed down from parents to offspring. C. A type of inheritance due to mutation caused by environmental factors. D. None of the above. The answer is A. An inheritance pattern in which the DNA sequence isn't altered but gene expression is. Question 3. If it were possible to clone Einstein, would the clones be equally intelligent? The answer is that clones may not be exact copies. Most traits are not governed by genes alone. There is an interaction between genes, environment, and epigenetic factors. Question 4. Is the following statement true or false? Epigenetic tags are not copied when the DNA is copied and passed on to daughter cells in cell division. The answer is false. In cell division, it is necessary to have the same genes switched on and off so that many cells carry out the same function in tissues. In sexual reproduction, most but not all of the epigenetic tags are removed in the fertilized cell. If you found this video helpful, do watch other videos on this channel. Thank you.